Welcome to the very first lecture video on neuroanatomy and physiology. This is the first of many to come. The focus of this particular lecture is all about nervous tissue. It's basically the types of cells there are and any subtypes and their basic functions. So here is a layout of how the nervous system is organized. Two major divisions: central nervous system that consists of the brain and the spinal cord. Peripheral nervous system consists of the nerves that connect the CNS, central nervous system, to the peripheral nervous system. One more set of structures that you could consider adding here are sensory receptors. Well, sensory neurons often serve as sensory receptors. So in that regard, it's already covered. Okay. So any further classification is where we could talk about somatic and autonomic divisions. Somatic is where the action, the sensation, or the response is all voluntarily controlled, or one is aware of these responses or these sensations. However, the opposite of that is autonomic division. Autonomic division is where you are not aware of these. It's involuntarily controlled. Breathing, heartbeat, all of these are involuntary functions. A good example of a somatic would be moving the hand from one place to another voluntarily, or from a hot stove, withdrawing the hand. So these are somatic movements. All right, so now autonomic is further organized into two subdivisions: sympathetic and parasympathetic divisions. Okay, so those are the parts of the autonomic division. What that means is these are all involuntarily controlled. We can add another extension to this, that is the enteric nervous system. Enteric nervous system is the involuntary supply specifically to the gut. The gut has two supplies. One is local, and that is what you're referring to as enteric. And then the other one is central. That is where you can think about autonomic supply. Okay, with this basic description of how the nervous system is laid out, let's now talk about these parts, where they are, and the names again. So here you're talking about the brain and the spinal cord, the two major organs of the central nervous system. Then you have the nerves, a number of them shown, and the enteric nervous system shown. Ganglia are groups of cell bodies in the peripheral nervous system, which we will talk about mostly in detail in the autonomic nervous system discussion. Okay, now we want to get into the tissue details. Tissue, specifically nervous tissue, is made of two types of cells. Neurons are nerve cell, and the other cells are neuroglia, or glial cells. Okay, neurons are the key type of cells, main type of cells that are responsible for the excitation, which is in response to the detection of a stimulus, or in responding to a stimulus in the form of generating a response. So those are functions. So it is basically processing information, providing response is how we would put it together. Neuroglia are the supporting cells. They support neurons. if you want to look at the number of neurons, you are looking at trillions of these, and then the neuroglia, their number exceeds that of the neurons. You're talking about 4 to 15 times more neuroglia than neurons themselves. Here is a classic structure of a neuron. This is actually a multipolar neuron. Okay, multipolar meaning that the cell body gives off many shootings. The smaller ones are dendrites. The main one is the axon. All right, so when you're looking at the multipolar neuron, we can also organize the neuron structure into its key parts. Cell body, also called as soma, is the control center of the neuron. This is where the nucleus is situated. This is where synthesis of cell supplies, including neurotransmitters, neuropeptides, all those things happen here, okay? And then the substances are carried into the axon, and you may have heard of exoplasmic transport. Okay, so it usually occurs in a forward direction, but there are instances where the substances can move towards the cell body. That's when you call it as retrograde transport, okay? Retrograde is the opposite of the forward movement of substances from the cell body to the axon. Okay, so now let's talk about dendrites. Dendrites basically connect this neuron to other cells, other neurons. Other cells could include muscle cell, gland cell, adipocyte, whatever it is. So basically you're talking about communication as the main function of the dendrites. They basically receive signals from other cells and allow it to arrive here into the cell body. Okay, so they receive the nerve impulses. That is the main function of the dendrites. Now, axon is the one that carries signal from the cell body away from the cell body, typically towards the axon terminal. Axon terminal, as the name suggests, is the end of the axon. And note that the axon terminal is branched. The branching can vary. You may have hundreds to thousands of branches here, or just a few. And then the very tip of the axon terminal is somewhat bulged. You refer to that as axon hillock. All right, so now what's the function of the axon? Carry the action potential or nerve impulse away from the cell body towards the very end of the axon. Okay, so those are basic functions, and we will look at them in relation to other topics that we will discuss. So here is another key area. You call that as the initial segment. Okay, so initial segment is where the origin of the action potential typically occurs. Okay, so this is a junction between the cell body and the axon. Okay, it has another name, axon hillock. Axon hillock, or initial segment, basically is where the action potential originates. Okay, so it starts here. Why does it start just here? Could be the question you have. Because this is where very high concentration of voltage-gated channels, sodium channels, potassium channels are concentrated. So this is a key area. All right. Now, the other details that you may be wondering about are these greenish coats here. This is myelinated axon. Myelin sheath is wrapping the axon in several places. In fact, it would wrap the entire axon, leaving tiny gaps in between. Those gaps are referred to as nodes of Ranvier. Okay, so the unmyelinated part of the axon is node of Ranvier. Note that the cell body and the dendrites do not have that myelin sheath, only the axon. Most neurons, most axons have myelin sheaths, so they're referred to as myelinated. Okay, a small percentage of axons do not have this myelin coat, and they would be referred to as unmyelinated. Okay, only a small percentage, about 2% of the axons are unmyelinated. Okay, so this is the anatomy of the neuron itself. Basically, it's histology because neurons are typically microscopic. Now, let's talk about neuroglia. What you do right away is to organize these into neuroglia that are in the central nervous system and neuroglia that are in the peripheral nervous system. Why? Because there are many kinds of glial cells. Okay, if you were to focus on the ones in the CNS, there are 4 distinct types: astrocytes, ependymal cells, oligodendrocytes, and microglia. We'll be brief about the role of these cells. Astrocytes, the star-shaped cells, are the ones in the greater number. Okay, most abundant ones are astrocytes. Their function is to provide an additional coat over the capillaries within the CNS. So capillaries have their endothelium, their basement membrane. On top of all of this, you would have another sheet that is laid by astrocyte. It's the cytoplasmic extension that reaches the capillaries and provide this additional layer. Why? Because in this manner, the substances that are in the blood won't be able to leak out by non-specific means. The specific means are a diffusion, osmosis, active transport, all these processes that occur at the cell membrane. Okay, so those happen as usual, but any leakage of unknown substances is avoided. So that is one function of astrocytes. The additional layer you have on the capillaries is called blood-brain barrier. Okay, almost all of the capillaries in the CNS have this additional coat, except in a few places. The example of that one place where capillaries lack blood-brain barrier would be choroid plexus around the ventricles. There is a reason why there is no blood-brain barrier around some of these capillaries. That's because these are the capillaries that are used to extract plasma and now brought into the ventricles, and that's what we call a cerebrospinal fluid. Okay? So with that, let's look at other functions of astrocytes. Besides producing this protective barrier, astrocytes also help maintain a normal status within the extracellular fluid, meaning any neurotransmitters that are released by the neurons are simply cleared. Okay, removed, so that there is no abnormal levels of other agents influencing neurons. Ionic concentration is maintained as well. So astrocytes are very important because they maintain the chemical composition of the extracellular fluid around the neuron, then they also provide physical support. Physical support because they have these cytoplasmic extensions. If you think of this as the astrocyte, it's labeled here. Do you see these cytoplasmic extensions? Those are the ones that simply provide physical support to the neurons nearby. So those are some important functions for astrocytes. Now let's talk about ependymal cells. Ependymal cells simply line the ventricles or the central canal. Ventricles and central canal are cavities. Ventricles are cavities within the brain. Central canal is the cavity within the spinal cord. They have a lining of cells around these spaces. This lining is what you call as ependymal cells. As I mentioned before, plasma is extracted from the nearby capillaries, and that is brought into these spaces, and then this fluid gets a new name called the cerebrospinal fluid. Who does that? Ependymal cells. Ependymal cells are the ones that steal plasma from these capillaries that lack this protective blood-brain barrier and then make CSF. So in order to give one function to ependymal cells, you simplify the function as ependymal cells make CSF. Okay, oligodendrocytes. What are these cells supposed to do in the CNS? Well, let's find oligodendrocytes. Do you see these round cell bodies here and here? And they have long cytoplasmic extensions. Those are oligodendrocytes. What they're doing is to add their myelin coats on the axons. One oligodendrocyte is capable of providing myelin coat around many axons. This one here is providing myelin sheath around this neuron as well as this one. And this one similarly is providing a myelin sheath here and on this axon. So that is how they add a myelin sheath. That's the function of oligodendrocytes. We have a different cell that does a similar function, similar thing, that is adding myelin sheath in the peripheral nervous system. That is going to be Schwann cell. We'll talk about it very soon. Microglia are the smallest of the glial cells. They are nothing but white blood cells, macrophages, that are found in the central nervous system. Macrophages are actually monocytes that are in circulation that migrate to tissues. Macrophages within the brain tissue are called microglia. Microglia, one cell shown here, they move from one place to another. So they're not fixed in one place. By being able to move, they would phagocytose debris or pathogens and basically clear these things from the extracellular fluid and the tissue within the brain and the spinal cord. So with this detail, let's focus on the glial cells in the peripheral nervous system. Schwann cells are the major ones here. If you're looking at Schwann cells, these are Schwann cells. Here, their main function is to provide myelin sheath. Each cell makes up one bit of myelin sheath. Do you see here? This is entire Schwann cell serving as one myelin sheath over one short segment of axon. And so if you're looking at a long axon, you would be looking at the lining of a number of Schwann cells. Okay, in some cases there are like 500 Schwann cells around one axon. Okay, again, they have this node of Ranvier. One interesting thing to remember is Schwann cells, because the entire cell participates in this myelin sheath, the nucleus and the other cytoplasmic content is all squeezed to the topmost layer. So the top layer looks bulged, whereas in the CNS, myelin sheath doesn't have that bulge because oligodendrocytes use their cytoplasmic extensions, not the cell body and the nucleus, to provide myelin sheath. The advantage of Schwann cell lining the axon is that these cells may remain in a place even if the axon were to be damaged. So I'm going to pretend that there is a cut here. What happens is it may be the axon that is cut off, but the Schwann cell is still in place. So as the axon shoots off its branches, one fine branch that fits through this space created by the Schwann cell can continue to grow and reestablish the connection. So that is where you can attribute another function to the Schwann cell. One, it provides myelin sheath. Two, it helps neuron regenerate, at least the axon regenerate, okay? Now, the last cell in the list, those are satellite cells. Satellite cells basically cover the cell body of a sensory neuron and the ganglia. Okay, so that is where you typically find. What do they do here is supposed to be very similar to the function of astrocytes, basically maintaining the extracellular fluid chemical composition, and the physical support is what I presume. Okay, so that is the end of our discussion of neuroglia and their functions. Here is the process of myelin sheath formation. This is one Schwann cell that you're looking at. The cell the nucleus of the myelin sheath is pushed to the topmost layer, and this is how the deeper layers look, very thin and concentric, whereas here it is kind of a bulge. Okay, so now let's do this. Let's classify the neurons in using two different categories. One is based on the structure, the other one is based on the function. Based on the structure, you already know about multipolar neuron. Okay, they are the most numerous, and you're talking about them as the motor neurons, multipolar neurons. Motor neurons are examples of multipolar neurons. Okay, we will soon talk about what motor neurons right here. Okay, we already know how they look also multipolar because many extensions are possible from the cell body. Most of them are dendrites. One specific one is the axon. What about the unipolar and bipolar? I have a slide here. In fact, I sat down and drew this unipolar and bipolar neurons. Here is the unipolar neuron. What you see is one cell body with the nucleus, and this extension here is just one extension, which then soon separates into two directions, and that is the reason why you call it as unipolar. Unipolar neurons are typically the ones you find as the general sensory neurons. Pain receptor, for example, thermoreceptor, those are unipolar neurons. What about bipolar neurons? As you probably guessed, it has two extensions, cell body right in the middle, one extension serving as axon, or you could call it as dendrite, the other extension serving as the axon. And the interesting thing is a bipolar neuron is commonly seen as a specialized sensory cell. Bipolar neuron that makes up the rod or the cone or a hair cells in the cochlea are examples of bipolar neurons, special sensory structures. Okay, so those are the three kinds of neurons based on the structure. Sometimes the unipolar neuron is also called a pseudo-unipolar neuron. Now, let's stick to the functional classification. This is important. Okay, we have three kinds of neurons based on the function. Afferent, note the letter A, and so afferent, these are sensory neurons. Okay, their direction of carrying sensory signals is from the peripheral nervous system to the central nervous system. So it's moving up towards the brain is the kind of message it gives. Afferent neuron is sensory neuron. Okay, so afferent neuron cannot be anything else but sensory neuron. Okay, now efferent neuron is the opposite. If you remember it that way, it originates in the CNS and it leaves the CNS and ends in the peripheral nervous system, typically on a muscle cell or a gland cell. Okay, mostly muscle cells. So efferent neuron is a motor neuron. Because it ends on the muscle, muscle involves movement, it is called motor neuron. Okay, association neuron is only found in the CNS. It's the one that connects other neurons together. Okay, bridges other neurons. It could connect an afferent neuron to an efferent neuron or other neurons within the CNS. Okay, so you're talking about them as interneurons in between neurons within the CNS. Very good. So you will see those in here. Let me start off with one example. This is a pathway that is shown. There is a lot of information here that you could talk about, but we will stick to just the recognition of sensory and motor neuron. Okay, sensory neuron here is shown in green. Okay, pick up signal, sensory signal here because it shows the tap and the water. You're thinking about cold or a heat that is being sensed by the fingertip where you have the sensory receptor, which is actually the sensory nerve ending, correct? And it's carrying signal. I want you to focus on the arrow moving upwards. Do you see that? That upward arrow is carrying the signal to the CNS, and that is basically showing the cerebral cortex. These details will be discussed later in relevance to other topic, but for now, the signal being carried to the CNS, this is spinal cord, and there are other structures that are not shown here, and now it is the cerebral cortex. So that entire pathway here is a sensory pathway. Okay, and now let's say the signal is processed and the response has to be given, and now you're looking at this red line. This is our motor pathway. Okay, it originates in the CNS, correct? It originates in the CNS, and I want you to look at the arrow here that is showing that this signal is moving away from the CNS, meaning reaching the peripheral nervous system. In here, it supplies the muscle in your palm, you see? So when it's hot, you may want to move your hand. If it's cold, again, you may want to move your hand. So that kind of signal is arriving through this motor pathway, originating in the CNS, ending in the PNS. So that is as much information as we need for now using this image. Okay, same thing here, but in a little bit more detail. This is the motor pathway that we followed earlier. What you're looking at is the neuron contacting the muscle. Do you see the muscle here in pink? Okay, and also the nerve ending is showing that bulge, synaptic knob. So all those details that you couldn't see before are magnified in here. Okay, and the junction between the neuron and the muscle is referred to as neuromuscular junction that we will talk about elsewhere in this chapter. Okay, so that's how far we want to cover in this lecture. The next one is going to be all about the electrical properties of the neuron.